Thank you, Adil. That's, I think, a great way to kick off the session and really shows how the science of correlative biomarkers can impact on the design of combination strategies, even if the biomarker is not being used very specifically to select patients for studies. So outstanding. So our next speaker is Dr. Ahmad Tarhini from the Cleveland Clinic, who's going to speak about master protocols for immunotherapy combinations.
Thanks, George. I'm grateful for the organizers for the opportunity to be part of this meeting. These are my disclosures. I'm going to start by giving an example about the drug development in stage 4 melanoma metastatic disease and how long it took to get where we are today. So for melanoma before 2011, we had only 2 drugs that were considered standard of care, dacarbazine and IL-2, that had FDA approval. At the time, 1-year survival was less than 25%. Between 1998 and 2011, we had absolutely no approvals, but a lot of work was being done in the clinic and in the lab, obviously. And then suddenly we had this explosion of approvals between 2011, 2015, 10 approvals, translating into a 1-year survival of over 70%. When we look at the durability of these benefits in terms of survival over time, where we stand today is 3-year overall survival with anti-PD-1 antibodies and in combination with anti-CTLA-4. About 50% of our patients would be alive at 3 years. Fast forward, the most recent update from the Keynote-001 study by Hamid at ASCO. This is pembrolizumab. If we look at the 5-year survival in the entire patient population. This phase 1 study, 655 patients, was 34%. The treatment naive was 41%. Major improvement, obviously. We have pushed the survival curve in a significant way upward, but it also means that about at least 50 to 60% of our patients still require additional treatment, and of course underscores the importance of accelerating drug development and investigation for these patients. The good news is, obviously, we have an ever-expanding understanding of the cancer biology and host immunology, and we have an, let's say, ever-expanding basket of checkpoint inhibitors and agonists and understanding the targets that can be targeted, and many ongoing studies, as illustrated by previous speakers today, that if we think about a PD-1, PD-L1 backbone, studies are combining immunotherapy with immunotherapy, target therapy, chemotherapy, and radiotherapy. So a lot of work being done, but obviously there are major challenges. So if we talk about the challenges facing immuno-oncology drug development today, our current classic model is a series of clinical trials testing 1 or 2 questions at a time in a single disease. This comes at a major cost in terms of time. Obviously, the financial cost. A lot of studies, maybe not too many studies, but basically competing studies. That's the biggest problem, is that many questions targeting a specific patient population, let's say PD-1 failures in melanoma as an example. So a lot of competing trials that are affecting our ability to finish those studies in a timely fashion. Most of our studies target patients in an unselected way in terms of mechanism or biomarkers or likelihood of benefit or understanding of the mechanisms of resistance before they go onto these studies. We have a huge population of screen failures where we don't really have adequate let's say, experimental options in terms of studies. So we're not thinking much about screen failures who, let's say, fail the classic, eligibility criteria for these studies. There is the group of those patients who may have rare genetic or, let's say, uncommon immunophenotypic subtypes that it's hard to target them and finish the studies in a timely fashion. And whether we have truly predictive biomarkers that can guide us in developing these studies. So obviously there is a major need for more efficient strategies that can test more approaches more efficiently in less time. And this is where master protocols could play a major role. So this could be a major methodologic innovation over the traditional approach of a series of clinical trials where we may have an overall systematic approach to a disease more efficient screening, increasing the speed of drug development and approvals. So we have major opportunities, obviously, to design and conduct master protocols for immunotherapy, building upon what is unprecedented, obviously, genomic and immunologic understanding of cancers, allowing us to incorporate precision medicine approaches, and allowing us to design mechanism-based trials, and allowing target and agent prioritization. These master protocols can allow a timely assessment of safety and clinical activity of multiple agents or combinations in parallel or in rapid sequences within, let's say, one major master protocol. I might argue that it would be probably important to take advantage of the NCI-sponsored cooperative groups in conducting such studies, taking advantage of a robust clinical trial infrastructure, allowing multi-sponsor trials and what can be easily, let's say, coordinated as a multi-stakeholder decision-making body, making these studies probably more feasible. So types of master protocols, there are three major types that, they're the common types, where most current master protocols are either belong to one of those categories or are, fall into one or two of these categories. So the first is the umbrella protocols, where these are meant to study multiple therapies in the context of a single disease, the umbrella. One example from a review article by Woodcock and colleagues in New England Journal of Medicine last year. So this is an example of an umbrella study which is meant to evaluate several subgroups within a conventionally defined disease. Patients are screened for the presence of a biomarker or other characteristic and assigned a stratum. Multiple drugs are studied in the various strata, and design may be randomized or may use external controls. One example of an umbrella trial is the NCI match, which, when we think about a single disease within the NCI match, this could be looked at as an umbrella study. As you know, NCI match is an umbrella trial to determine whether treating cancers according to molecular abnormalities is effective. It is targeting advanced solid tumors, lymphoma, myeloma. Each can be thought of as a separate umbrella within a basket of multiple diseases. Of course, this involved DNA sequencing for actionable mutations. And then these are, let's say, multiple treatments are targeted based on these gene abnormalities. So NCI match obviously is an exploratory, non-comparative study. The primary endpoint is a tumor response as a primary, and also progression-free survival. The sample size is relatively small, 35 patients per sub-study, looking for signals that potentially can be taken into larger, let's say, randomized phase 2 or phase 3 studies, but could serve as a model. So one might envision a anti-PD-1, anti-PD-L1 backbone umbrella study, where patients are screened based on the presence of certain biomarkers and allocated to certain strata or combinations, building upon these anti-PD-1, PD-L1 monotherapy. The next type of master protocol is the basket study. So this is meant to study a single therapy in the context of multiple diseases or disease subtypes, basically the basket. Again, from the review article by Woodcock and colleagues in New England Journal of Medicine, this is described as it involves multiple diseases or histologic features. Participants are screened for the presence of a target and entered, and could contain multiple strata that test various biomarker drug pairs. One example is the BRAF V600 study was mentioned earlier today. This is a basket trial to evaluate the efficacy of vemurafenib involving multiple non-melanoma cancers with BRAF V600 mutations. It is a phase 2 non-comparative adaptive trial using Simon two-stage design, and the response rate was the primary endpoint. Of course, this was published in 2015. Another example from the IO world is the ECHO 202 or Keynote 037 study. This is the study which was a basket study of multiple diseases that evaluated the efficacy of the combination of pembrolizumab and epacadostat, IDO inhibitor. The primary endpoint was overall response rate. and involved, as a basket, multiple solid tumors and diffuse large B-cell lymphoma. The sample size within each arm was about 25 to 42 trials, and this is the study from which, obviously the phase 3 ACO 301, which I'll mention a little bit about it later, came out basically based on the promising, phase 2 data from this basket trial. The third type of a master protocol is the platform study, and this is meant to study multiple therapies in the context of a single disease in a perpetual manner, with therapies allowed to enter or leave the platform on the basis of a decision algorithm. A potential design also here proposed by Woodcock and colleagues of a platform trial. In this case, this is a kind of a platform trial ongoing over time with no fixed stopping date and governed by a master protocol that envisions adding and dropping strata. So here patients are, let's say, screened based on biomarkers, and they enter a certain stratum. In biomarker A case, we have investigational drug 1, 2, and a standard of care. Drug 1 potentially continues until it becomes successful. So this arm is dropped, and then it replaces the standard of care when this becomes the standard of care. And then you add another investigational drug. This one probably becomes negative, and it is dropped, and others are added, and so on. So obviously there are other types of adaptive designs possible, and different decision rules, statistical or biomarker signatures that can be used in designing these studies. One example of an adaptive platform study is obviously the I-SPY2 study, which is an exciting study, not only being the adaptive platform design, but also being a neoadjuvant study. And I know there's a lot of excitement among this group based on previous discussions in terms of the value of neoadjuvant testing, especially in drug development. So I-SPY2 is an adaptive platform phase 2 randomized study. It is a neoadjuvant study, patients with breast cancer, locally advanced disease. It involves a number of biomarkers, including hormone receptor status, HER2 status, MAMA print risk score, defining 8 genetic subgroups. And the primary endpoint is the pathologic complete response rate. And here, I'd like to kind of, again, stress probably the potential importance of neoadjuvant therapy, probably early on in guiding further drug development, taking into account maybe lessons learned from the epacadostat, data where we had a phase 2 study that was positive base response rate, went forward. Should we have done more, such as neoadjuvant testing, learning more from the biology? So one example in terms of neoadjuvant immunotherapy study is one of the studies that we conducted with ipilimumab. So basically the design here is patients with bulky disease. They get a baseline excisional biopsy, which is mandatory because, diagnostic, you need to get an excisional biopsy, you have enough tumor that can be diagnostic and also can be used for research. In this study, patients were treated for 2 doses of ipilimumab, basically 6 weeks, and then underwent definitive surgery where, again, we have access to a, let's say, large amount of tumor, both diagnostic and for research. In parallel, can collect, blood biospecimens before, after, and then long term. Something that probably can be done as we go forward, especially when we're talking about a high bar to beat, let's say in melanoma, the anti-PD-1 immunotherapy effectiveness. So the advantages of neoadjuvant therapy, clinically, obviously, the improved clinical outcome. There's a separate question, we're interested in this, but also experimentally in drug development in the IO world is the ability to evaluate clinical and radiologic as well as pathologic responses, guiding the future decisions, and of course the access to the tumor and blood before and after, investigating the mechanisms as well as the biomarkers, potential predictors of response going forward. Not to present our data, but just to give some example about the wealth of data we were able to generate from such a study. In the tumor, we were able to show that at 6 weeks only, there was a massive infiltration of CD8 T cells, after only 2 doses of ipilimumab, and where we found a significant association with response based on the density of the CD8 T cell infiltration within the invasive margin. doing gene expression profiling, we found a pro-inflammatory gene expression profile where we were able to separate patients in terms of the relapse risk, relapse-free survival, into very high risk of relapse or low risk of relapse based on their ability to, based on their tumor's expression of certain immune-related pro-inflammatory gene signature. In the circulation, working with Lisa Butterfield, we're able to monitor regulatory T cells, myeloid-derived suppressor cells, effector T cell response, and also be able to understand the impact of ipilimumab, early on treatment and long-term, up to 1 year, on these different, let's say, regulatory as well as effector immune elements. So this wealth of data, and there's other things that we've published in this area, including, the toxicity risk and so on. If used early on in drug development, I think it can guide us better going forward before moving into phase 3 studies. This is a slide courtesy of Jason Luke, which is an exciting study. This is BMS CA028-001. It is meant as an adaptive design biomarker, let's say, driven study where patients who failed nivolumab from multiple tumors are stratified into multiple combinations here based on a biomarker signature. So this is the markers that they are interested in is CD8, PD-L1, LAG3-IDO, CSF1R, FOXP3 GITR, and KP46. And they came up with an adaptive, let's say, decision mechanism where based on the likelihood of response, let's say, into any of these combinations, the patients get stratified. I think it's a study, while it is phase 1, and the primary endpoint here is basically just, kind of a modest endpoint looking at, let's say, the number of patients with baseline tumor biopsy samples with more than or equal to 30% tumor content. Basically, it's a modest primary endpoint, but it's meant to collect mechanistic data. And I think it serves as a good model going forward as we think about the biomarkers where we can stratify patients into potential combinations. So considerations going forward for master protocols, trial designs. Obviously, important to think about the, as others have mentioned earlier today, histology specific or histology agnostic, various tumor types. The study endpoints, what is the primary study, the important study endpoint for these master protocols so we can make decisions to go forward into phase 3, the response rate, the PFS, PCR, neoadjuvant testing, randomized versus historical controls. The careful thing about the eligibility criteria early on, making sure these are representative eligibility criteria of the patient population going forward. Interim analyses and stopping rules, biomarkers and enrichment strategies. Obviously, the statistical analyses and thinking very early on in terms of registrational intent and discussing with the FDA, early on as such, major investments are made in master protocols. Challenges, obviously, there are a number of challenges, some of which are, first of all, meaningful short-term study endpoints that may allow adaptive designs. The right biomarkers that may allow adaptive designs as well. Collaborations across competing pharmaceutical companies. Logistical and operational considerations, and obviously funding and cost, all of which need to be think of as we design these studies. Going back to the example of epacadostat and pembrolizumab in melanoma. So this is, this came from the ECHO 202 study. It was a basket study, and it was positive. The overall response rate was 55%. So, these are exciting waterfall plots. And at the time, I think all of us supported, going forward into a phase 3 study. And these are the outcome was shown earlier today. Response rate with the combination epacadostat with pembro was 34%. Absolutely no difference in terms of PFS or OS. Hazard ratio was 1. Obviously, something went seriously wrong in interpreting, the results of the phase 1 study. So what lessons can be learned? So we're doing a master protocol. We have, promising arms, going forward into phase 3. Some of the thoughts. Number one, response rate was the primary endpoint. And this is, obviously clinical radiologic should we mandate, central radiology review for some of those, when it's such a major commitment or investment going forward into phase 3, potentially could help, in ensuring that this is, accurate data. Single arm versus historical control, prior to phase 3, single arm based on historical controls. Should we do randomization? Of course, it's more investment in time and effort, but at least in melanoma, where again we have a high bar with anti-PD-1 to beat. Now it's becoming harder, really, to kind of take a 50% response rate into phase 3 immediately. So I think thinking more and more, at least in our area, in terms of randomized phase 2 testing, the eligibility ensuring that it is representative, a patient population. In other words, if we have in melanoma, we have the M1A, M1B, M1C, and M1D. Ensuring that we are representing all of these patients, all these populations in the early development before we move into phase 3. Right, those were discussions in whether it should be 100 milligram BID for epacadostats or 300 BID, and did we do the right dose? And therefore, obviously, careful PK/PD testing early on is very important. And biomarkers and PCR, again, I'm a big fan of neoadjuvant therapy, and potentially really, we have discussion with the NCI, and Elad Sharon is here, in ECOG doing a platform neoadjuvant study, something we'll be discussing for a while. So recommendations going forward, and this is my last slide, is obviously thinking very early on in terms of the right design, depending on the questions going forward. Study arms that may provide options for screen failures. These patients don't have many options, and I think will improve accrual into these studies. The importance of obtaining tumor tissue in these studies to investigate biomarkers and resistance pathways. Standardized biospecimen acquisition and storage. Obviously, this is Lisa's topic, which we'll discuss at length. Considering neoadjuvant evaluation, collaboration and coordination between pharma, NCI, FDA, academia, community oncology programs, taking advantage of the NCI-sponsored corporate group infrastructure, guidance to community sites to allow broader participation, patient and healthcare provider education, and obviously rewarding participation and teamwork, especially if we want to make these massive protocols successful. Thank you.